All right, welcome everybody. The Office of Programs to Enhance Neuroscience Workforce Diversity within the National Institutes of Neurological Disorders and Stroke is pleased to welcome you to the Open Stage monthly webinar series. This is a targeted outreach effort for our diverse neuroscience community to learn about NINDS programs and network with NIH and NINDS program staff. Each webinar features a different program, division, or cluster within NINDS. This is all for kind of the goal of increasing the representation of diverse groups in our portfolio. So this month, we are very excited to get an overview of NINDS biomarker development and validation from NINDS program officers and awardees. Before we get started, I just have a few housekeeping items. If we could go to the next slide. Thank you. So just a few meeting reminders. I know you're familiar with Zoom, but all attendees are muted. So please submit your questions using the chat function only. And a reminder that this meeting is being recorded and it'll be transcribed and archived on our website. You can enable closed captioning under the captions or live transcript at the bottom of your screen. Details about where to find this archived version will be posted on social media and on our listservs. We will send an email. I will send an email after the meeting with the link to the recording, with the slides, with all of the resources shared. So you don't need to worry about that. You'll get a copy of the slides and all the information after the meeting. So please do stay connected with us. Feel free to email us if you have any questions about the webinar series. And with that, I will pass it off to Dr. Taylor-Burds to get us started.
And I will unmute. Thank you, Anna, and thank you everyone for joining us today. We're really excited to do this webinar in particular because we have two fabulous guest speakers with us today. Dr. Fred Korley, who's an associate professor of emergency medicine at the University of Michigan, and Dr. Maria Diaz-Ortiz, who is a MD PhD student at the University of Pennsylvania. And we're going to introduce them more later, but to get us started, I want to go through just a little bit of an overview to get everybody on the same page about some of the work around biomarker development and validation, terminology, and things we'll be talking about later in the discussion with our panelists. So I'm Carol Taylor-Burds. I'm a program director for the Neuroscience Translational program at NINDS, our Division of Translational Research. And with me is Dr. Adisa C. Liu, who's a health program specialist in the Division of Translational Research, as well as Dr. Christine Swanson-Fischer, who is the program director for the Accelerating Medicines Partnership for Parkinson's disease in our Division of Neuroscience. I think one of the themes you'll hear from across this webinar is that this is, biomarker work really spans basic science to clinical use, and we have biomarker work happening across the Institute, across programs, and so this is really an interdisciplinary research effort. So just to get us all on the same page, I want to make sure, we're all familiar. What is the definition of a biomarker? So it's defined as a characteristic that can be measured as an indicator of normal biological processes, pathogenic processes, or response to an exposure or intervention, including therapeutic interventions. Right, and this is a definition that is a NIH FDA biomarker working group, the best glossary that we're going to talk about a little bit more. And in addition to that, what a biomarker is, you'll also hear us talking about concept of interest. So what's the biomarker actually represent? What's the pathogenic process or the normal biological process that biomarker is an indicator of? And then how do you measure the biomarker? So what's the detection method? And that might be the assay or an imaging protocol, a detection algorithm, and so on, right? There's a lot of different types of biomarkers, both in terms of the modality as well as the use cases. And then talking about use cases, context of use is a really important term in regulatory considerations and something that we talk about a lot in the funding opportunities that we have in our translational program, because this is really describing how the biomarker is going to be used in a particular situation, whether that is as a drug development tool or in clinical practice. And that context is critical because it tells you what do you need to know about how that biomarker is going to be used in order to be fit for purpose in that case. So at workshops, at conferences, you'll always hear people talking about we need more biomarkers, and what does that really mean? And again, it depends on the context, right? Because in some cases, we need more biomarkers to understand disease etiology, the mechanisms, understanding the pathophysiology of the disease. We need biomarkers for use as tools in therapeutic development. And we need biomarkers for use to make decisions in clinical practice. And so these different contexts are important to consider when we're talking about we need more biomarkers. So when we're talking about the basic mechanistic side of things, you might start off with a question, identifying a biomarker or candidate biomarker that's associated with a disease or condition. And we're going to have a nice case example of this from Maria, who's going to be talking about some of her research in Parkinson's in this mechanistic space, and then moving on towards translation. And then what is that biological process that the biomarkers reflect? So you might need to understand more about the disease, the mechanisms between what the biomarker is actually related to, what pathogenic processes are there. And then how's the biological process related to the etiology of the disease condition? Is it driving disease progression? Is it an associated secondary complication, and so on? And so when we think about this, we really think in terms of we need more biomarkers to generate and test hypotheses about the disease etiology. So you can use biomarkers to help figure out what's happening within the condition or the disease. And we also need to understand the disease etiology in order to identify good candidate biomarkers for use in clinical trials and clinical practice. And so this is really iterative, building on knowledge in both directions. So because of the complication around using biomarker to mean a lot of different things, back in 2016, the NIH and FDA developed the BEST resource, or the Biomarkers, Endpoints, and Other Tools resource, to help classify biomarkers in terms of their use cases. And so they have several of these. So diagnostic, I think everybody's pretty familiar with, monitoring biomarkers, pharmacodynamic response biomarkers, predictive biomarkers, prognostic biomarkers, safety, and susceptibility risk. And I'm not going to go through these in detail now. All these links will be available in the slides if you want to take a look and think about them more. But if you think in terms of how are they used in therapeutic development, they can be kind of grouped into 5 broad categories. So diagnosis, again, pretty straightforward. Target engagement, so pharmacodynamic response biomarkers are often used to determine if the intervention is hitting the biological pathway or the mechanism of action that is the therapeutic interest, and trying to determine whether the dose is sufficient to elicit an adequate response or make dosing decisions. And then patient stratification is really important to help improve the efficiency of clinical trials by enriching patients with individuals who are more likely to benefit from a particular therapeutic, or more likely to have the condition that's therapeutics being developed for. And then monitoring progression, how's the disease changing over time? Does the biomarker change in response to the intervention over time? And safety toxicity, can the toxic or adverse reactions be detected or predicted before clinical symptoms or irreversible damage occurs? So when you look in terms of when are these biomarkers used in therapeutic development, in general, the pharmacodynamic response biomarkers are particularly helpful in that pre-IND, preclinical research and early phase 1 clinical studies, when you're trying to determine dosing decisions and show that your therapeutic is actually hitting the mechanism of action. And then prognostic, predictive, and susceptibility risk biomarkers are all particularly helpful for patient stratification in those later stage phase 2 and phase 3 clinical trials. And then diagnostic biomarkers, obviously selecting the right patient population, and monitoring and safety can really be used across the spectrum from early preclinical work through post-market surveillance. And then finally, if we think about using biomarkers in clinical decision-making. So again, these same categories of biomarkers, thinking about when and how they're used. You might have susceptibility risk biomarkers that would help identify individuals who might need additional screening or are likely to develop a particular disease condition. Identifying the right individuals, what the diagnosis actually is, determining if someone is more likely to develop a specific condition or have a more severe form of a disease, predict which treatment is going to be the best for that patient or that individual, and then whether or not the therapeutic is actually responding, or the person is responding to the therapeutic. And finally, again, monitoring and safety really throughout the therapeutic intervention and assessments. And so finally, in terms of discovery and development, there's sort of three broad categories you can think of: biomarker development, validation. Obviously, this is an iterative process, and it's a little more complicated than this, and it depends exactly on which biomarker context of use, how much evidence is needed. But you can think in terms of starting with discovery and development. Does the biomarker reflect the process for the proposed context? Can it be adequately measured? Analytical validation, really assessing that the detection method that you're using to measure the biomarker is reliable and can be trusted to give you the results that are accurate enough to be interpreted. Clinical validation, where you're doing those large clinical studies to determine how you are actually going to take the results from the biomarker measurements and make those interpretable and meaningful for making decisions at the individual participant level. And these definitions and categories are really built off of the FDA's biomarker qualification program. And the biomarker program has the FDA's biomarker qualification program is a drug development tool program to help get qualified biomarkers. And so qualification is the conclusion based on a formal regulatory process that within the stated context of use, a medical product or development tool can be relied on to have a specific interpretation and application in medical product development and regulatory review. So just to point out again, NIH and NINDS has research funding opportunities across the whole spectrum, from disease mechanisms and early discovery work. And that's mostly within sort of the division of neuroscience and your standard R01s and R21 mechanisms. We have our translational program, which is the one I lead for the division of translational research, and this is to get biomarkers validated for use as tools in clinical trials or clinical practice. The small business program has a parallel way of funding these, and we have a few programs that are small business focused as well. And then our division of clinical research that funds clinical trials and comparative effectiveness research. It also has some ancillary biomarker validation and development funding opportunities as well. So these links are available, and I'd encourage people who are interested in learning more to reach out to us or to anyone within their program director for their area of expertise to learn more. And these are the specific programs for funding opportunities for the NINDS biomarker program that I mentioned. And we do webinars twice a year. So we're encourage anyone who's interested in potentially applying for one of these to check out these resources. The past webinar is posted here. We'll be doing another one in April. And do let us know if you want to discuss your project. All right, and so with that, I am going to switch it over now. Give me one moment here. And we are really excited to have Dr. Fred Korley and Dr. Maria Diaz-Ortiz join us. I'm going to stop screening so I can introduce them properly. And all right, so before we get started, so Dr. Maria Diaz-Ortiz is an MD PhD student, as I said before, at the University of Pennsylvania in the lab of Dr. Alice Chen Plotkin, where she received her PhD in 2021 and led and published an article in Science describing how GPNMB confers risk of Parkinson's disease through interactions with alpha-synuclein. She and the Chen Plotkin lab are now extending that work to investigate if GPNMB is also a candidate target for therapeutic development. And at the same time, Maria is also working on characterizing Alzheimer's disease plasma biomarkers in diverse patient cohorts while completing her medical school training. In addition, we have Dr. Fred Korley, who, as I said, was an associate professor of emergency medicine at the University of Michigan. His research is focused on the development of novel diagnostics and therapeutics for traumatic brain injury. And he's the principal investigator of 2 federally funded multi-center research studies investigating the use of biofluid-based patient stratification biomarkers for use in clinical trials and for monitoring individual patient responses to promising neurotherapeutic agents. He is also the principal investigator of a NINDS-funded phase 2 adaptive design clinical trial. And Dr. Korley has 2 patents for biofluid-based biomarkers for brain injury detection and outcome prognostication. I'm not going to go into his list of awards, but I do want to mention he is also a member of the National Academies of Science, and he's also a standing reviewer, which we greatly appreciate, for the NINDS NSDA study section, where our biomarker validation studies, some of our biomarker validation studies are also reviewed. So with that, I am going to ask Dr. Korley if he can start off by telling us just a little bit about some of his research and what's going on in the TBI field and the need for biomarkers and what's being done in the space currently.
Thank you very much for the introduction, Carol, and thanks to you and your team for the invitation and putting up this great webinar. See, there are lots of participants, and thank to everyone for joining. So I'm going to focus primarily on clinical validation of biomarkers, and especially focus on the TBI space, the traumatic brain injury space. And I'm going to discuss a number of different studies we're conducting just to illustrate the different questions we think about when you're thinking about clinical validation of blood-based biomarkers. So, and I'll talk about a number of different contexts of use and try and keep this to 10 minutes. So to start off with, let's talk about diagnostic biomarkers. These are biomarkers that allow us to diagnose patients with a condition or help us in diagnosing patients with a particular condition. And as you may or may not be aware, the FDA recently cleared the very first set of blood-based biomarkers for detecting brain health. These biomarkers are GFAP and UCHL1, and they were cleared by the FDA in 2021 to be measured on a point-of-care platform. And this is really exciting because the initial indication is that these tests are supposed to help us with figuring out which suspected traumatic brain injury patients need a CT scan. So we see a ton of traumatic brain injury patients in the emergency department. Each year, we see about 5 million of them. About 80% of them get CT scans. 90% of the CTs are all negative. So there are lots of patients who are getting CTs who don't need CTs. And the problem with getting a CT scan is that it exposes patients to radiation. There is some time associated with waiting to get the CT and then waiting for the read, and that also increases their length of stay in the emergency department, not counting the costs to the healthcare system. And so these blood tests have been cleared so that we can use them as screening tools. So you get a blood test, and if someone is positive, then you get a CT scan, and if someone is negative, then you don't need a CT. But even though there's been a lot of work that's been done up to the point of getting these biomarkers cleared, there's still additional work that needs to be done before we can use them in the clinical space. And one of the main issues that we have, and that's why we've not started using this test in our emergency department yet, is that we don't know how it's going to work in our population. So I work in Ann Arbor, and we're not a major urban center, and so our patient population is a little bit different. For example, we see a lot of elderly patients. We see a lot of elderly patients who come because they've fallen, and so it's really important to understand how the biomarker is going to work in our particular patient population before we start using it. And so when we had funding from the Department of Defense to study 2,000 of our patients who are coming through our emergency departments and see the diagnostic accuracy of these biomarkers in our particular pragmatic cohort. And so what we do is every morning, we send a list to the lab of patients who came in the night before, who came in for a trauma, who had a CT scan, and who had blood samples. We collect their blood samples and we test them for these biomarkers. And we're almost up to 2,000 patients. One thing I can tell you is that compared to the published studies, our patients are on the average 10 years older than patients in published studies. And so what we're doing is really, really important. We're also finding out that there are some patients who present super early. We're able to see them and get blood samples in less than an hour from when they've had their injury. For a lot of clinical trials or a lot of the prospective studies that were used to validate these blood tests, patients need consent before you get blood. And so most of the time, these studies draw blood samples about 8 hours or so after injury. And so if you just use those results and start using that test clinically, you may not know how the test will perform if you get the blood samples, within an hour or so of injury. And so those are all the nuances that we're trying to tease out in our pragmatic study. And then we'll come up with an algorithm for how we can use this test, within our population. And then, and actually one other thing that we need to answer is that we clinicians don't make decisions in a void. So yes, a biomarker can have good diagnostic accuracy, but the question is the biomarker telling me something I don't already know? And that is a question you have to answer as part of your clinical validation work, that we currently have decision rules that need no money, no testing. You just ask patients some questions to help you figure out whether they need a CT scan or not. And before we start using our biomarker, we want to be able to demonstrate that this biomarker is doing better and telling us things that we would not have known by just talking to the patient. So that's about studies to do in terms of for validating a diagnostic biomarker. In terms of studies to validate prognostic biomarkers, so these are biomarkers that can help you predict the future from today. So for example, when a patient comes in with traumatic brain injury today, can you look in their blood and be able to tell how well they're going to do neurologically 6 months down the line? And so in collaboration with our colleagues who run the TRACK TBI network, which is a large observational study of traumatic brain injury patients, we looked at these two biomarkers that have been FDA cleared to see if it can help predict the future, to see whether based on blood samples drawn on the day of presentation, we can figure out which patients are going to have a good outcome or which patients are going to have a poor outcome. And, we definitely were able to demonstrate that, these biomarkers are really powerful and that they not only can reliably predict outcomes in patients with more severe traumatic brain injury, but they also add additional information to the other risk predictors that are currently available and that are used clinically. We also have been interested in seeing if we can predict stroke risk, especially now that we're able to measure these brain injury biomarkers. I think it's a brand new world to be able to see if we can do a better job of understanding brain health. I've heard so many people say that my family member was fine and then all of a sudden they had a stroke. And I have just never really believed that it happens that quickly. I don't think that stroke happens just all of a sudden. I think that before stroke actually happens, not for all patients, but for a lot of patients, there probably are these silent ischemic events, these small strokes that happen that are subclinical and do not show symptoms. And there are many small ones that happen before the big one. And these brain injury biomarkers may be able to help us pick up these small brain infarcts that are happening along the line. and we did a study to investigate this. And part of the reason part of the reason for highlighting this particular study is also to let people know that there are available biorepositories run by both the NINDS and NHLBI that you can leverage to answer important clinical validation questions for biomarkers. So to do this study, I reached out to NHLBI, and they have a public website where you can request for samples for previously completed studies. And there's a study of patients who are diabetic, who were randomized to a number of different treatments that I'm not going to get into, and then they were followed for outcomes. And so we selected patients who had never had a stroke within that cohort, and followed them up for 7 years, to figure out who had a stroke. And we requested the samples from NHLBI. The process was very simple, and we measured a biomarker called neurofilament light chain in those patients. And what we found was that if your neurofilament light chain level was in the very high category, you were almost 10 times more likely to have a stroke in the coming 7 years than if your neurofilament light levels were super low. And so, this opens up a new avenue for these biomarkers, because potentially, in the future, when you go to your primary care doctor's office, they may be able to measure some of these brain injury biomarkers. If the level is really high, it may mean that maybe we need to target a lower blood pressure for your blood pressure control, or we need to be more aggressive with controlling your glucose, or more aggressive with controlling your cholesterol, and so on. So those are prognostic markers and how they can help us with improving care. I want to talk about predictive markers. I think predictive markers are really awesome because they are markers that can help predict how well a patient is going to do, respond to a particular treatment. And we've been doing a study of hyperbaric oxygen in traumatic brain injury. And as part of that study, we're collecting blood samples. And we're going to look at the baseline blood samples and see if they help us to identify patients who respond favorably to hyperbaric oxygen therapy than patients who don't receive hyperbaric oxygen therapy. And so for the clinical validation of predictive biomarkers, it's going to be best to do those with ongoing clinical trials. And so if you have a marker that you think has the ability to identify patients who respond favorably to one particular treatment, then it's going to be important to partner with one of the ongoing clinical trials that are studying that particular condition and collect blood samples and examine them. The last group of biomarkers I'm going to talk about is monitoring biomarkers, which allow you to monitor a patient's disease state. And with that, longitudinal measurements of those biomarkers are going to be important. And we do think that these brain injury biomarkers that were noticed that have been recently validated may be important for monitoring patients' response. And so there are a number of ongoing clinical trials, where we're collecting blood samples and collecting the samples serially and examining the patients serially and looking to see if the patient's characteristic matches with the level of the blood-based biomarker. And I think my time is up, so I'm going to pause now and maybe take questions later.
Sounds great. Thank you so much, Fred. That was fabulous. Maria.
I'm going to take this from, hi, everybody. I'm Swanson-Fischer, Program Director at NINDS. I'm so excited to be part of this panel today, and I'm so happy that Maria and Fred could join us as we're talking about how to develop, discover, and validate biomarkers for for neurological disorders and stroke. So Maria, I'm going to focus with you a little bit backwards. So Fred was talking a lot about he's been able to get these biomarkers validated in the clinic, but I'm going to take a few steps back because you've been in the laboratory of Alice Chen-Plotkin doing a little bit more early development of these biomarkers. And I wonder if you could talk a little bit first about the unmet need for developing biomarkers in Parkinson's disease and how that's kind of helped you guide your research over the last few years.
Absolutely. So as some of you may know, Parkinson's disease is a huge clinical problem. It's essentially the #2 most common neurodegenerative disease, second only to Alzheimer's. And right now, clinically, these patients are diagnosed based on the presence of clinical criteria, essentially whether these patients have a tremor, they have rigidity, they have postural instability, and the absence of red flags that point us in another direction, like really fast clinical progression. However, we do not routinely use any biofluid-based biomarkers in the diagnosis of Parkinson's disease. There is the presence of dopamine transporter imaging, but this is only used as supportive criteria for the diagnosis of Parkinson's disease. So there's a huge unmet need for diagnostic biomarkers. Secondly, in Parkinson's disease, we have this phenomenon where about 90% of Parkinson's disease cases are sporadic and only 10% of them are familial or with a known genetic cause. So this 90% group is very heterogeneous because they could have many contributing risk factors that contribute to the development of Parkinson's disease. And what this heterogeneity manifests as is that if you take very well characterized patient cohorts like the PPMI Parkinson's disease cohort, and you sort of segment them into thirds, like the top third progressors versus medium progressors and slow progressors, you can see that over the course of 5 years, there's a third of patients that will have no progression of their motor symptoms and are basically stable. And this is a problem for clinical trial development, because if you're trying to show that a therapy has a disease-modifying effect, but you have no idea whether this individual patient was going to progress or not, how can you actually tell that your therapy is going to work? So there's also a very large need for prognostic biomarkers. And so in our group, when I sort of joined the lab, we were interested in this question of, can we identify novel biomarkers for the diagnosis and prognosis of Parkinson's disease in an unbiased way? And we also decided to look at plasma or blood-based biomarkers rather than CSF, just because clinically plasma is much more accessible. If we can find something in the plasma, it is more easily translated into clinic rather than having to ask patients to perform a lumbar tap, which, if done properly can be okay, but can also be quite uncomfortable for patients. And so we essentially started off with a cohort of Parkinson's disease patients, about 95 patients and 45 neurologically normal controls from our own institution. And we quantified over 1,000 proteins in plasma samples from these individuals using an aptamer-based platform. Essentially, aptamers are modified nucleic acids that have higher affinity for protein that allows us to pull down the protein and then quantify these aptamers using microarray technology. So you can do this in a very high throughput manner. So by doing this, we essentially identified about 140 candidate biomarkers that sort of enrich for Parkinson's disease in our discovery cohort. But again, when it comes to biomarkers, the trick is validation, validation, validation. So we then proceeded on to use actually an NINDS resource. We used another cohort that was available to us through the Parkinson's disease biomarkers program. And this cohort was composed of individuals with Parkinson's disease and neurologically normal controls again. But they proceeded from 2 different clinical sites, again, to try to capture higher variability that you would see in the real world. And so by applying similar methods to this discovery cohort, we sort of found out that 4 of these biomarkers replicated their associations in our discovery cohort. But then in order to sort of kind of verify whether these markers were true markers, we sort of asked if they also allowed us to prognosticate or determine which patients were going to develop cognitive decline over the next couple of years. And we ended up finding out that 2 of these biomarkers were really good at predicting cognitive decline, not only in our discovery cohort, but also in our validation cohort, and not just with one cognitive measure like the dementia rating scale, but also looking at progression in terms of consensus diagnosis. So progressing from cognitively normal to mild cognitive impairment to dementia.
That's great. And so you've taken this, I think that the results from your study are kind of bi-directional, because you're able to see now GPNMB as a potential risk marker for Parkinson's disease. And if I gathered right from your paper, also potentially associating with disease severity. So you could track that as a potential therapeutic target. But at the same time, what I thought you did was so elegant is that you started trying to understand how is GPNB interacting with the disease pathology. And I wonder if you could just briefly talk about how you went about trying to elucidate that, and how you've been able to, on the one hand, now start to interrogate the function of GPNMB, but at the same time, my understanding is now you're starting to also look on the other side of how we can attack this as a therapeutic target for Parkinson's disease.
Of course. So GPNMB was of interest to us because while I was working on this initial unbiased biomarker-based project, we sort of ended up finding out that GPNMB was one of these markers that associated with a more rapid trajectory. So patients who had higher GPNMB in their plasma also tended to have a faster cognitive decline. And there was a lot of evidence in the literature pointing towards a role for GPNMB in Parkinson's disease. Essentially, genome-wide association studies looking at various risk loci and risk of Parkinson's disease had identified a locus near GPNMB, but not in GPNMB exon, as a recurrent locus that increased risk for Parkinson's disease. And other studies had shown that patients with Parkinson's disease had elevated levels of GPNMB in their substantia nigra when you looked at cross sections. And so we sort of, it's funny because in our paper, it's like the last figure, but we sort of started with some of the biomarker work since we already had a lot of resources available to us. And we did find out that, it looks like patients with Parkinson's disease had elevated GPNMB in their plasma. And when we looked at their CSF, we could see essentially like a genetic locus and protein level association, or an expression quantitative trait loci, whereby the SNP that was associated with increased risk of Parkinson's disease, patients who had the SNP also had higher GPNMB in their CSF. And so this led us to believe, or to hypothesize, that this GWAS risk locus increases risk for Parkinson's by mediating expression levels of GPNMB. And so for me, being a bioengineer, my first thought was like, I want to engineer cells to remove GPNMB. What happens if we knock out this protein in terms of, alpha-synuclein, since alpha-synuclein is thought to be like a major modulator of like Parkinson's disease. And so what we found is that essentially cells where we knock out the GPNMB had a very distinct phenotype in terms of their alpha-synuclein. It looked like they have lower alpha-synuclein expression. When we looked at them in an unbiased way with RNA sequencing, we also saw a lot of gene expression changes, but one of the hub genes of these gene expression changing modules was alpha-synuclein, pointing to an interaction between these two proteins. So we hypothesized that this link between the two might actually be a physical interaction. And so we performed co-immunoprecipitation experiments, found out that these proteins do physically interact. And then knowing, or sort of, there's this belief in the field that alpha-synuclein behaves as a prion-like protein and sort of spreads throughout the brain, right? Affecting the substantia nigra first and then spreading to other interconnected brain regions. This is sort of supported by the Braak hypothesis and evidence from postmortem brains showing pathology early on in the substantia nigra, and with patients who have more advanced disease, they do have less involvement, but still some involvement of cortical areas. And so we sort of looked at experiments that sort of recapitulated some aspects of the cell-to-cell spread. And so in our cells where we knock out GPNMB, when we treat them with exogenous alpha-synuclein that is capable of seeding pathology, these cells take up less alpha-synuclein and they form less pathology, suggesting that this GPNMB alpha-synuclein interaction might be important for this cell-to-cell spread that is sort of a hallmark, we believe, of Parkinson's disease. And furthermore, if we take cells that normally don't internalize alpha-synuclein, and now we introduce GPNMB, we add the ability to internalize alpha-synuclein, sort of suggesting that GPNMB might be both necessary and sufficient for the uptake of alpha-synuclein. And so, you know, the question is, what are we doing in lab to sort of get this over the hump and into translation? And, you know, essentially GPNMB is a cell surface protein. So initially we thought that it might be a cell surface receptor for alpha-synuclein and its internalization. However, it's also cleaved and secreted as a soluble protein, which is probably what we're able to detect in plasma. Regardless, this is very exciting because it means that, you know, any alpha-synuclein GPNMB interaction is happening in the extracellular space, which means that we can target it with antibody-based therapies. And so right now in lab, there's another excellent graduate student who is doing the work of trying to block this internalization with antibodies and having some, you know, exciting data. We're also doing some animal work, some animal studies with mice that lack GPNMB, and we're injecting them with alpha-synuclein preformed fibrils to look at whether knockdown of GPNMB affects the spread. So stay tuned for those results in the future.
Fantastic. Thanks so much, Maria. Appreciate it. I'm going to put it back to you, Carol. I think we can move into the panel. I think we've got a bunch of questions in the chat as well.
Sounds great. And thank you again, Maria. Adisa, do you want to run the Q&A for us?
Absolutely. So we do have a couple questions. The first question is, are there NIH or DoD funding opportunities for interventions for mild to moderate TBI or CTE?
And I guess I can take that one. So I would encourage you to look at the NINDS funding opportunities site. You can see a list of active programs. Also feel free to reach out to me or Encini Umo, who is the program director for the CTE portfolio at NINDS, but just shoot me an email and I can point you in the direction if you need assistance with that. We have quite a few funding opportunities, so at any one time, it's always good to see what's out now.
Absolutely. Thank you, Carol. The next question is, in terms of grantsmanship, a biomarker could be applicable to several classifications of biomarkers. Were there several boxes that need to be checked to show you have validated the biomarker for that class? Is it still advantageous to mention how it can also be used, aka future directions, or is it encouraged to validate for more than one classifications?
Right, that is a good question too. So for the translational biomarker funding opportunities that we showed in the slides before, and that Dr. Korley also was one of the reviewers on, we do ask that you're very specific about the context of use because the program goals is to help develop the evidence necessary for getting that biomarker into use or towards qualification. And so you want to be pretty specific and think about how does your study design match which context of use you're proposing. In the R01 space, that might not be as important. You can think more broadly in terms of what are the different use cases. And I know that the biomarkers that Korley mentioned, you know, a number of those are the same, you know, markers like NFL and GFAP, UCHL1, but they're being used in different contexts and validated in different contexts. So maybe I'll turn it over to Fred and see if he wants to comment more.
So I agree with everything you said, Carol. I think it's in an R01 grant, it's not unreasonable to propose validation of different kinds of biomarkers if you're leveraging one study. I think for the U01 translational piece, it's going to be hard to justify validating, both like a prognostic marker and a diagnostic marker. That's going to be a little of a stretch. I can see how you could, if you're looking at a prognostic marker, you could have a number of different contexts of use, right? You can have a marker that helps you prognosticate on the day of presentation, and then see if it also does a good job, for, when they're seen later in clinic. And that would not be too schizophrenic. It seems like it's, around the same theme. But if it's, two different types of biomarkers, I think that's going to be hard to put together in one grant.
And you've had a lot of experience looking at these in different contexts and validating them. It's a pretty serious effort, right, on the part of the study design and how you go about validating them for those use cases.
Exactly.
Absolutely. Thank you so much. The next question states, there are a variety of biomarkers, for example, plasma biopsy and CSF for the diagnosis of synonucleopathies such as PD and LBD. Are these views as unreliable at this time? Are these viewed as unreliable at this time?
Maria, do you want to take that?
Sure, I am happy to. I think, there are currently none that are FDA approved, but I think, further validation is probably needed at the time. And I think, it's important, especially if you're looking at like synucleinopathies, to have like association, with some sort of postmortem data as well, especially if you're looking at synucleinopathy, like you want to make sure that you're not just doing an association with like a clinical phenomenon.
Right. And I think we're getting at this the stage where there's a lot of consensus around that there probably should be a panel of biomarkers or a multimodal approach. And I know that in the works, there are synuclein PET tracers, still very early on, and the data is coming out showing a high affinity for multiple systems atrophy, but not so much of the other synucleinopathies. So I think there's a lot of work to be done, and that's why I'm so excited about the work that Maria and others are doing, especially also within our Parkinson's disease biomarkers program and with my colleagues at the Michael J. Fox Foundation.
But yes, there are certainly a lot of like very exciting things coming out the pipeline, especially with some of the RT-QuIC assays.
Absolutely.
Thank you all. The next question is for Dr. Korley. How can we differentiate a peripheral protein metabolite slash metabolite is neuron specific or glial specific? Any experiments you recommend?
So that's it's always challenging. There are a number of proteins that are brain enriched, but doesn't mean they're not found elsewhere. And there, some proteins that are just in the nervous system, both central and peripheral. So sometimes it's hard to distinguish. Looking at exosomes is one approach. Cell-specific exosomes is one approach for trying to determine whether your analyte is specific to a particular cell. And, so long as they're tagged for that particular cell, you can interrogate the cargo. And then if they're found, in exosomes from a particular type of cell, then you could be more comfortable saying that, that particular analyte you're measuring, the one you measured is, from that cell type.
And there, I think we get back into the sort of basic science side of things where we really need to understand how reliable the markers for the vesicles are and how easily you can enrich them and make sure that you're measuring from the sources you think. So again, that basic science informing clinical and vice versa.
Thank you. The next question is for the panel in general. Do you know the latest on EEG biomarkers being explored in relation to neurodisorders and their use in clinical practice?
Do either of you want to take that? If not, I will give a general answer.
Yeah, that's a very general question. It's because, you know, the EEG markers for different domains, traumatic brain injury, you know, post cardiac arrest, seizures, they're all sort of different. So I don't know that there is, you know, you can state one type of EEG biomarker that is being used for in relation to neuro disorders in general. I don't know. Do you have a?
Yeah, no, I mean, I agree.
Specific?
You're talking about basically a modality for how you're measuring something, but the actual biomarker would be a particular electrical feature or features from the EEG, and those would be the biomarkers, and that would get into how's it being used and what context. So I know within neurodevelopmental disorders and epilepsy, obviously, there's a lot of interest in developing biomarkers using EEG, but we think about that more in terms of sort of the detection method rather than the biomarker itself, if that makes sense.
Thank you for your answers. The next question is, does the level of the biomarker correlate with Parkinson's disease severity or change with treatment? Also, this particular biomarker is also associated with many cancers, including myeloma. What is the percentage increase in protein relative to baseline levels?
So I think this question is referring to GP&MB, so I'll go ahead and jump in. So in our paper, we described an association between GP&MB plasma levels and Parkinson's disease motor severity as measured by the UPDRS Part 3 score, which is essentially a score of like motor dysfunction. And yes, GP&MB, so the M in GP&MB stands for melanoma. So it has been well studied in cancers, and there are some associations between GP&MB and various cancers. Whether, you know, GP&MB can be a marker of like treatment response, that remains to be seen, because we currently don't have any disease modifying therapies. And we also did not perform a study in Parkinson's disease patients pre and post dopaminergic therapy. So that's a good question. The answer is we don't know. And then in terms of the percentage increase relative to baseline levels, it was slight. It was not, you know, like twofold or something really large where we had a lot of separation. So, you know, when thinking about whether this would be a great diagnostic biomarker, probably not, possibly because of some of the other issues raised, right? It seems like it might not be a very specific biomarker for Parkinson's disease, but if it, you know, serves as a lead for furthering our mechanistic understanding of Parkinson's disease, you know, I'd say that's still a win.
Thank you for your answer. So the next question states, with regards to suppressing / inhibiting alpha-synuclein pathology / spreading, does this correlate to improve functional outcome in PD? At times, the pathological improvement does not match improved outcome.
Maria, you want to take that?
Yeah, sure. I'm happy to jump back in. So yeah, again, this is a very good question. I think, you know, there is evidence for alpha-synuclein being causal in PD from, you know, genetic cases where you have either like a missense mutation in alpha-synuclein or duplication of the alpha-synuclein gene. These patients do have altered alpha-synuclein folding or expression, and, you know, this is thought to be causal, but we still, right, therapies targeting alpha-synuclein are still in clinical trials. So whether this results in an improvement in functional outcome, whether that be motor or the development of cognitive symptoms, still remains to be seen. But again, I think a very good point, you know, with the advent of anti-amyloid therapies in Alzheimer's disease, it's certainly a question that we should be posing.
Absolutely. Thank you for your answer. The next question states, in cancer, biomarkers / panels have not necessarily proven so valuable, which gets to the point that quantification, to the point of quantification. The value of any biomarker is dependent on the ability to accurately measure them. Are the projects ongoing along these lines? If the assay sensitivity and specificity is not good enough, then what?
Kara, do you?
So, so I think again, it depends on what context and which markers we're talking about here. So maybe I think, you know, getting to the sensitivity and the specificity of the assays, and this is a good question, right? Because we didn't talk so much about the detection methods and how accurate those, and I know you both have experience talking about that as well. So maybe I'll ask Fred to comment first, because I know you've actually specifically looked at different assays and evaluated their sensitivity and specificity, especially like the point of care devices.
Yeah, I mean, analytical validation is like its own rigorous set of work that needs to be done before you start thinking about clinical validation. You have to be 100% sure that you're able to measure your analytes with accuracy and precision. And those all mean, you know, important things that, you know, can be discussed. But you're absolutely right that, you know, GFAP from manufacturer A is not necessarily the same as GFAP from manufacturer B. You know, the numbers may mean completely different things. And so like you cannot necessarily think about an assay or a marker outside the detection method. And frankly, most of the time when the FDA clears a marker, most of the time they're clearing, you know, the detection method plus the marker.
And that's a great point too, that we didn't have time to talk about, but there are different regulatory pathways. So the devices or the assays will get regulated and reviewed through CDRH, whereas the biomarker qualification program is specifically for drug development tools, and that has a different regulatory pathway. I think with, you know, we didn't talk about the analytical validation as much, but again, is you have a good candidate biomarker that's really going to potentially be useful, but you can't measure it accurately enough to use it for that context. And so again, there's iteration and development to improve the detection method as well as the evaluating the biomarker itself. And Maria, did you want to add anything? No, we're getting close to time too, so maybe just one or two more questions.
Yeah, we do have a couple more. I think the next question will be for Carol. How much preliminary data is required for the R61, R33 grant?
I think there's no short answer to that, but if you want to reach out and have a draft aims page, we're happy to talk about what's the best funding opportunity for you. So just email me. Thanks.
Great. And maybe a last question. What is your opinion on using biomarkers that may show a decrease from baseline, indicating pathology/disease, as opposed to always looking at biomarkers that increase relative to baseline?
I mean, I think, again, this depends on the context, right? So depending on what the question is and how it's proposing to be used is really sort of the other information we need to think about here. don't know if anybody else wants to comment on that.
Yeah, so I think, it's reasonable to do also in an unbiased approach, right? So when we were doing our unbiased screen of biomarkers, we looked at any association, meaning are the markers elevated in Parkinson's disease or are they decreased in Parkinson's disease? And when I was talking about our 4 candidate biomarkers that replicated across cohorts, some were in the direction of elevation in Parkinson's disease and some were lower in Parkinson's disease. So there is the possibility, but again, it just comes down to what question are you asking?
And I just want to quickly point out that clinicians are used to using biomarkers that go down. So for example, vitamin D levels, right? They're a type of biomarker and they go down. So it's not unreasonable to have a biomarker like that.
Good points. I know we are out of time. I really want to thank you all again for joining us. This was great. I'm so glad we had you here to talk with us. I will turn it over to Anna for closing remarks.
Thank you so much. Thank you to our amazing panel, for all of you for attending live. We had amazing questions, a lot of questions from the audience, so that's great. Again, this will all be recorded. I'm sorry we were having issues with captioning, but we will have the full transcript available on our YouTube page when we post it. So we will share all of that. In the meantime, again, we will send all registrants an email once the recording is up, and we'll include the slides and all the resources. And please do sign up for our listserv so that you hear all about future open stage webinars and other resources. So with that, thank you so much. Have a great rest of your Monday.